Protein synthesis. What is protein synthesis? Protein synthesis is an essential component of memory formation. An A2 regulates the synthesis of new proteins through at least two different estrogen receptors. Mediated mechanisms, the classical genomic pathway and the rapid non-classical activation of cell signaling pathway. Protein synthesis, transcription, and translation. Transcription. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. DNA safely and stably stores genetic material in the nuclei of cells as reference or template because it carries the same information as DNA but is not used for long-term storage and can freely exit the nucleus. Although the mRNA contains the same information, it is not an identical copy of DNA segment because its sequence is complementary to the DNA template. Transcription is carried out by an enzyme called RNA polymerase and a number of accessory proteins called transcription factors. Transcription factors can bind to specific DNA sequence called enhancer and promoter sequence in order to recruit RNA polymerase to an appropriate transcription site. Together, the transcription factors and RNA polymerase form a complex called transcription initiation complex. This complex initiates transcription and the RNA polymerase begins mRNA synthesis by matching complementary bases to the original DNA strand. The mRNA molecule is elongated and once the strand is completed, synthesized transcription is terminated. The newly formed mRNA copies of the gene then serve as blueprints for protein synthesis during the process of translation. What is RNA? RNA is a single-stranded, not double-stranded, like DNA. RNA is short, only one gene long, where DNA is very long and contains many genes. RNA uses the sugar ribose instead of deoxyribose in DNA. RNA uses the bases uracil instead of thymine in the DNA. The stages of transcription are initiation, elongation, and termination. Initiation. After RNA polymerase binds to the promoter, the DNA strands unwind, and the polymerase initiates RNA synthesis at the start point on the template strand. Elongation. The polymerase moves downstream, unwinding the DNA and elongating. The RNA is transcribed 5 to 3. In the wake of transcription, the DNA strand reform a double helix. Termination. Eventually, the RNA transcript is released and the polymerase detached from DNA. Synthesis of RNA transcription. Initiation. Promoters signal the initiation of RNA synthesis. Transcription factors help eukaryotic RNA polymerase recognize promoter sequences. Synthesis of an RNA transcript. Elongation. RNA polymerase synthesizes a single strand of RNA against the DNA template strand, adding nucleotides to to the tree end of the RNA chain. As RNA polymerase moves along the DNA, it continues to untwist a double helix, exposing about 10 to 20 DNA bases at a time for pairing with RNA nucleotides. Synthesis of an RNA transcript. Termination. Specific sequences in the DNA signal termination of transcription. When one of these is encountered by the polymerase, the RNA transcript is released from the DNA and the double helix can zip up again. Post-termination RNA processing. Most eukaryotic mRNA aren't ready to be translated into protein directly after transcribed from DNA. mRNA requires processing. Transcription of RNA processing occurs in the nucleus. After this, the messenger RNA moves to the cytoplasm for translation. This cells adds a protective cap to one end and a pair of AS to the other end. This both function